A pleasant morning to everyone. In behalf of my team, we would like to welcome you on our short discussion about cell
Cell
injury.
injury.
And
and
adaptation.
adaptation.
So let's begin. So for our first topic, cell injury, we'll talk about how it happens and the types of injury and what causes it to happen. Cell injury happens when cells are stressed so severely that they are no longer able to adapt, or when cells are exposed to inherently damaging agents or suffer from intrinsic abnormalities. Cell injury have two types of cell injury. The first one is the reversible injury. Cell injury is classified to be irreversible if the injured cell can regain its homeostasis and go back to its normal state. An injury that can be reversible if it can be reversed by oxygen. An injury that on its starting phase are considered to be mild injury, tapos kapag naibalik yung oxygen, babalik na rin yung cell to its normal state. Yung reversible injury is caused by lack of oxygen or blood flow of the cells. Hallmarks of reversible injury. Reduced oxidative phosphorylation with resultant depletion of energy stores in the form of adenosine triphosphate. When the mitochondrion doesn't take oxygen, it cannot produce ATP. ATP is used by other cells to work on their jobs. Therefore, if there is no ATP, it will result to cell damage or even die. Cellular swelling, when there is a change in ion concentrations and water influx. Features of reversible injury. Cellular swelling. It happens when cells are incapable of maintaining ionic and fluid homeostasis and is the result of failure of energy-dependent ion pumps in the plasma membrane. There would be a swelling if the cell cannot control the influx sodium ions and water and efflux of potassium ions to the cytosol. Fatty change. It occurs in hypoxic injury and various forms of toxic or metabolic injury that is manifested by the appearance of the lipid vacuoles in the cytoplasm. Meaning, when the cell that is involved in the metabolism of fat, like liver cell, are damaged, it will unable to metabolize fat, then the small vacuoles of the fat will be accumulated by the cytoplasm. Next is irreversible injury. Cell injury is classified as irreversible if the stimulus persists or it is severe and compared to the reversible injury will result to cell death already. Irreversible is caused by immunological responses or viral infections. Mechanism of irreversible injury. Inability to reverse mitochondrial function. Even after the correction of the original injury, there is still a lack of oxidative phosphorylation and ATP generation. Development of profound disturbances in membrane function.
Here are some causes of cell injury. Hypoxia is the loss of oxygen supply and is an extremely important and common cause of cell injury and cell death. It is different from ischemia because ischemia is the reduction of blood flow due to obstruction in the blood vessel. Anemia, which is the reduction of oxygen capacity of the blood, may also result to cell injury. Physical agents, mechanical trauma, temperatures, radiation, electric shock, and sudden changes in atmospheric pressures. All of these can cause cell injury as temperature affect the ability of cell to function well, and other physical agents such as mechanical trauma can damage the different structures of the cell. Chemical agents, including simple chemical substances such as glucose and salt, can alter the vital functions of the cells. Their harmfulness depends on the concentration, like when oxygen in high concentration becomes severely toxic, and the chemical reactions of some chemical agents are harmful. Infectious agents. It refers to viruses, to bacteria, to multicellular parasites. They cause wide range of cell injury because some produce toxins and some compete with other cells in nutrients. Immunological reactions cause the defense system to attack other cells or cause immune system to release a flood of chemicals that can cause hypoxia. Example, anaphylactic reaction to foreign protein or drug, and autoimmune diseases like lupus. Nutritional imbalances cause cell injury as cell cannot properly function when there is excess or insufficient amount of nutrient. Genetic defects causes cell injury due to changes in amino acid and congenital enzymatic deficiencies. Alteration of single amino acid can cause disease like sickle cell anemia, while congenital enzymatic deficiencies due to alteration at DNA level causes error of metabolism. Let us move on to the general considerations of cell injury. These are the most forms of cell injury. Morphological changes can be observed due to disturbance of critical biochemical systems. First, first lesion will develop at molecular level, then it causes structural changes at an electron microscopical level. Then light microscopic lesion develops. When extensive, it can now be seen with the naked eye, which is the gross lesion. Cellular response depends on type of injury, duration, and severity. For example, low doses of toxins may cause reversible response, while larger doses of toxins may cause irreversible response. Result of an injurious stimulus depends on the following: type, status, adaptability, and genetic makeup of the injured cell. Same injury has different result depending on the cell type. For example, striated skeletal muscle in the legs can endure complete ischemia for 2 to 3 hours without irreversible injury, whereas cardiac muscle dies after only 20 to 30 minutes. Intracellular systems that are exposed to attack of injurious agents are cell membrane, anaerobic respiration, synthesis of enzymic and structural proteins, and preservation of the integrity of genetic apparatus of the cell. Structural and biochemical components of a cell, closely related to one another, that the injury at one side leads to wide-ranging secondary effects. For example, impairment of aerobic respiration disrupts the energy-dependent sodium pump in the membrane that maintains ionic and fluid balance, altering the intracellular content of ions and water, resulting to rupture of the cell. These are several common biochemical mechanisms in the mediation of cell injury and cell death. ATP depletion. ATP is generated by first, the major route, which is oxidative phosphorylation of ADP, and the second, the glycolytic pathway, which can generate in the absence of oxygen. But ischemic and toxic injury causes ATP depletion because of lack of oxygen supply or normal functions are deranged. Lack of oxygen or generation of oxygen-derived radicals. Both contribute to cell death and cell injury. Byproducts of mitochondrial respiration, the cell generate energy by reducing molecular oxygen to water. During this process, small amounts of partially reduced reactive oxygen are formed. They have unpaired electrons that make them highly toxic molecules that can damage lipids, proteins, and nucleic acids. So oxidative stress, a condition associated with cell injury. It forms when there is an imbalance between free radical generating and free radical inactivating systems. Stimuli such as radiation, chemicals, induce damage through activated oxygen species, while ischemia causes cell injury by reducing the oxygen supply of the cell, resulting to cell injury. The cell is composed of low concentration of calcium in the cytosol, mitochondria, and endoplasmic reticulum. But due to ischemia and toxins and the release of calcium from mitochondria and endoplasmic reticulum, cytosolic calcium increases, in turn activates a number of enzymes with harmful cellular effects. So intracellular cytosolic calcium activates the following harmful enzymes. Phospholipases causes membrane damage. Proteases cause breakdown structural and membrane proteins. ATPases cause accelerated ATP depletion, and endonucleases cause nuclear chromatin damage. Consequences of increased intracellular calcium in cell injury. So injurious agent allows the net influx of extracellular calcium, and along with calcium release of mitochondria and endoplasmic reticulum contribute to cytosolic calcium, which activates ATPase, phospholipase, protease, and endonuclease. Defects in membrane permeability. Compromised by pathogens that release bacterial toxins and infectious viral proteins. Several types of cell of immune systems such as cytotoxic lymphocytes have the capacity to attack other cells, physical and chemical agents. Changes in membrane permeability may result to loss of ATP and calcium activation of phospholipases. Mitochondrial damage. Mitochondria are most likely to get damaged by increase of cytosolic calcium, which was discussed a while ago, oxidative stress, the imbalance between free radical generating and free radical inactivating systems, breakdown of phospholipid through phospholipases. The damage caused of phospholipases is at the membrane of mitochondria. This results to damage in the formation of high conductance channels, or also referred to mitochondrial permeability in the inner mitochondrial membrane. Due to the formation of these pores, protein gradient across the mitochondrial membrane disappears, preventing ATP generation. Leakage of cytochrome C, which leads to the activation of apoptotic death pathways, because cytochrome C is an important soluble protein in the electron transport chain. So here, an injurious agent is introduced to the mitochondria. The damage will ultimately result in the formation of mitochondrial permeability transitions. Then due to that, the protein gradients across the mitochondrial membrane disappears. The damage will also result in leakage of cytochrome C, which will lead to secondary apoptotic effects.
Next is free radical cell injury. Free radical cell injury, known as the injuries that are induced by the free radicals, particularly those activated oxygen species. As we all know, free radicals are known to be the molecules that only have one electron in outer orbital. Mechanism of cell injury. Lipid peroxidation of membranes. The presence of the oxygen may cause peroxidation of the lipids in plasma and organellular membranes. Cross-linking of proteins. The free radical acts on the sulfhydryl bonds of proteins in it and activates the sulfhydryl enzymes. DNA fragmentation. When free radical reacts with thymine in nuclear and mitochondrial DNA, they produce single-stranded breaks.